welcome back. This is chapter 14. I know we're jumping backwards here. This is Alice, and we are going to talk about translation and the genetic code. We started it in transcription. I want to do a little bit of the details that we look at in bacterial translation in part 1 of this video, and then part 2 we're going to look at how eukaryotic translation differs. She always leaves as I start talking. Here we go. So here we're going to do in video 1, I want to talk about prokaryotes, translation, what the structure and function of a ribosome, the structure and function of tRNA. You know this. I'm going to add in some other new pieces of information. I will point out that our understanding of this has changed pretty dramatically in the past 5 years. So I'm not holding any of the old stuff against you, but I want to point out that this is one of the cool places in class that we can really, and as we go forward, probably everything we talk about, we're at the forefront of our understanding here. So you're learning what we think is happening now, but this is not like in Bio 1 when we're like water bonds to other waters. We've known that for a long time. This is stuff that we are learning now. And our understanding, therefore, changes pretty rapidly. So I want to tell you what we know now, and then give you some modifications. A little bit about translation. Translation is that part of the flow of information in living organisms where we take something from an mRNA transcript and convert it into a protein or a polypeptide. We talked about in lecture 13, so I think you've done 20 in between, but lecture 13, when we talked about transcription, we talked about how the information from DNA was converted into mRNA, and that differed a little bit based on whether you were a bacteria or a prokaryote or a eukaryote like us. Either way, at the end of that process, the mRNA needs to be transported to a ribosome. So what do you remember about ribosomes? And this is a picture of a ribosome here. What do you remember from Bio 1 and from microbiology? Because the process of translation utilizes the catalytic, so the fact that this is a ribozyme or a ribosome that facilitates chemical reactions to happen faster or with less upfront energy, so that we can catalyze the formation of peptide bonds, join amino acids together with covalent bonds. The ribosome is made out of proteins and rRNA. It is not an organelle. There is no membrane bound around it. It does not sit away from the rest of the structures in the cytoplasm or the cytosol. It is a machine. It is not a container, whereas organelles are really containers. Its job is to catalyze peptide bonds, and to do that under the instructions of the code in the mRNA that is coming from the DNA instructions, so that it consistently builds the same order of amino acids and ultimately the same functional proteins. Sorry, we're still running across the keyboard here. tRNA is the type of RNA that reads the codons. It has an anticodon on it, and it's spring. I'm sorry, the bird is going to be bonkers today. Please be patient with the fact that I have a 23-year-old toddler running in and out of my keyboard. The tRNA reads the codons on the mRNA. Basically, it has a 3-nucleotide anticodon on it, and it brings the appropriate or the cognate amino acid to the assembling polypeptide. If some of this doesn't make sense, let's review it. I think what happened with DNA on the exam, and DNA replication in particular, is that everyone kind of knew it already. You've had it so many times that I think fewer people, you spent less time reviewing it than you needed to. I want to avoid that mistake here. This is something you have heard in multiple classes, but this is tricky stuff, and it's complicated. And a lot of these words probably vaguely make sense to you. I want to make sure that it's more than vague, that you really understand what I'm saying. So when I talk about the ribosomes being made of protein and rRNA, not being containers, but being machines, that you remember that these are folded structures inside of the cell itself. And inside those ribosomes, there are places where interactions and actions take place. From micro, and if you didn't have micro, so this may not have been something you discussed in Bio 1, from microbiology, you talked about the fact that the size and the structure of the ribosomes differ between prokaryotes and eukaryotes. This is part of the way that we know that things like mitochondria have a bacterial origin, because the ribosomes inside of mitochondria are 70S ribosomes. They are ribosomes that look more like a bacterial ribosome. This S stands for a sedimentation factor. When we spin them in a centrifuge, they're smaller. They sediment out at a different rate than a eukaryotic 80S factor. I think you also learned that inside of each ribosome, there are 3 spots. Some of you, if you learned this in high school, they tend to do 2 spots. There are 3 spots where an action takes place inside of the ribosomes. These are, we now know, powered by the ribosome itself. This may be different. It is different than what you learned in Bio 1. I'm pretty sure that it's the ribosome itself, not enzymes inside the ribosomes, that are driving this process. When I look at ribosomes, I can see there is a large and a small subunit. This is really a large piece and a small piece. In this picture, this is the small piece, and this is the large piece, small piece, large piece. In bacteria, the large piece is made out of a 23S RNA. That means its sediment value is 23. There's a 5S rRNA. It's very tiny, and 33 proteins that are associated with those rRNAs. In eukaryotes, it's a larger structure overall. The large subunit is made of a 28S rRNA. It means it sediments out at a different rate than the bacterial large subunit, and a 5S rRNA, and a 5.8S rRNA with fewer ribosomal proteins. The basic structure is still the same. I get this little blob and a big blob, and when they come together, I get 3 places inside of these blobs where a particular reaction takes place, or a few reactions take place. tRNA is going to help us inside the ribosome read the mRNA so that we bring the right amino acid. tRNA has a really classic cloverleaf structure. The fact that when this was discovered, this is held together with, these are representing hydrogen bonds. So maybe this is an A and a T and a G and a C. This is a single-stranded molecule of RNA that then folds in this very characteristic shape. These shapes are fairly well conserved across different groups of organisms. When I did my graduate research and I was looking at trying to find ways to tell if my insects that I was working with were part of the same family or different families than other groups, one of the ways I looked at them was their structure of their tRNA and how their tRNAs differed from each other. So when I say it's conserved meaning it's not subjected to a lot of evolutionary pressure. This was a good way for me to basically classify them in their wider group, the coding in here. This structure is held together in a cloverleaf shape with a series of hydrogen bonds. On one side of this picture is an anticodon. This is the I represents, it can be anything, it doesn't matter. The 3' side of the molecule itself is where the amino acid that is the cognate to this code actually binds. Cognate means the match to that code. In this picture, it is a tRNA of tyrosine. This end would be the amino acid tyrosine, and this would be the anticodon that matches the codon on the mRNA for tyrosine. There is on the 5' end, there's consistently a guanine with a phosphate group stuck to it that kind of caps off this end. I want to introduce a new enzyme to you. I know that you were just thinking, wow, we haven't had a new enzyme in like 15 minutes. So here's one. This is my favorite enzyme in the history of all enzymes as far as I'm concerned. Its name is amazing. This is amino acyl tRNA synthase, which if we break it down, A's means it's an enzyme. It's a type of enzyme that synthesizes things, so it builds bonds together. The molecule that it works on is a tRNA and an amino acid. This enzyme's name is amino acid and tRNA synthesizer. Guess what it does? It puts them together. So every time you have a tRNA and you use it, it donates its attached amino acid to the growing polypeptide chain. We now have an empty tRNA. It immediately goes to an aminoacetyl tRNA synthase, and that reloads it. We say it recharges it, and it puts an amino acid, the appropriate amino acid, back on the tRNA. We call that charging. So our tRNA is either going to be charged, it has an amino acid, or uncharged, it does not have an amino acid. Oops, I drew on it again. I always forget that these don't layer over my drawings. I took this from a totally different textbook. Your textbook uses this picture to show everything, which I don't think is particularly clear. Aminoacetyl tRNA synthase is an enzyme. It grabs the amino acid in its active site. It's a synthesizer, so it needs ATP as a source of energy. It takes an ATP and hydrolyzes it. It breaks off the phosphates here. And when it does that, it grabs onto a tRNA. The tRNA binds to this site. It's a little bit wobbly. So at any tRNA that kind of fits will match with this particular cognate amino acid. It smooshes them together. It catalyzes the formation of this bond right here between the tRNA and the amino acid, and then it releases this amino acid attached to its tRNA as a charged tRNA to be used again during translation. It reloads the tRNA. When we put this all together, and we had a little bit in lecture 13 and a little bit in lecture 14 about translation, we're going to do a process using ribosomes, mRNA, tRNA, amino acids, and we're going to build a growing chain of amino acids. We're building a molecule, so we have to follow the same kind of pattern that we have every time we build a molecule. We have to do something to start it. We're going to initiate it. Then we're going to continue that process and elongate, make the process longer and longer and longer and longer. And then we're going to have some kind of ending that says that this is the end of the process and no longer keep building. I would compare this back to PCR as well. So at this point, by the end of this lecture, you should have 4 different stories: DNA replication, transcription, DNA polymerase, PCR, and DNA polymerase chain reaction, and translation, where you have these 3 processes: an initiation, an elongation, and termination. I highly recommend that you build out that chart that I gave you in lecture 13, add translation to it, and find the places where these things are similar and different. We're building amino acid chains now. We have not used amino acids to build in any of the other processes. We're gonna need, in order to initiate this process, a ribosome. But the ribosome comes disassembled. It's kind of like from IKEA. It comes separated. We got to put it together to make the process happen. It has a small subunit and a large subunit, and they are not together when we start this process. We're going to need some energy, and it has to be energy in the form of GTP. ATP is what we've been using for most of the processes we've talked about, but this is an adenine and 3 phosphates on it. Guanine is also a purine. It is the same kind of structure as adenine, and it also has the same functionality as an energy molecule. You've probably seen it in other places in textbooks. I don't know if anyone has pointed it out to you. ATP is molecularly more often used, but there's no reason why we can't use a guanine and 3 phosphates. I had asked my professor at one point in molecular biology and biochem why we use ATP preferentially, and he felt it was just an evolutionary chance that ATP was the one that happened to happen first, and GTP had nothing wrong with it. It's just used less commonly. We're going to need some initiation factors, which are going to be enzymes or proteins that are going to be available for us to initiate the process. We're going to need our first tRNA. It is going to be methionine. I always mispronounce it, but it is going to be a particular version, MET, that is going to bind with our start codon. And we need our transcript, right? We need our instructions. Not everything in this picture is here because we're only initiating for this part. Inside the ribosomes, there are catalytic subunits. What that means, I made this all red because we have, this matters. It means there are places where reaction takes place. There is A. A is the arrival site. The charged tRNA comes in at A. It attaches the new amino acid to the growing polypeptide at A. This is hard to remember. I don't know why, but it's really hard to remember for me and absolutely everyone that we arrive and attach. So I put all the A words here at the, it's really called the amino acidyl site, but the A site. The P site, so this is going to be 1. Step 2 is going to be the P site. The peptidyl site holds the polypeptide. This has changed, our understanding of this has changed very recently. This peptidyl stood for peptidyl transferase, which was the enzyme that we thought did this. We don't think that it does that anymore, but it is there. It is where we actually hold and elongate. It's going to come out the top here, or the bottom in this picture. This is where the polypeptide, the chain of amino acids, is actually going to be held. And then the exit site, the E site here, is where the uncharged, it gave its amino acid to the growing polypeptide chain. This is where the uncharged tRNA exit the ribosome. It's going to be grabbed by amino acidyl tRNA synthase and recharged. There are 3 initiation factors that bind to the small subunit, and then it binds to the mRNA. So this small subunit of the small piece of the ribosome is hanging out on its own, doing its own thing. We do think that in most organisms, ribosomes are kind of specific to the protein that they make. So it's waiting for its own particular kind of mRNA. I don't have a mechanism here for this. We just think that that is what is happening. In bacteria, when we grab on to the piece of mRNA with the initiation factors helping, it is looking for a sequence of nucleotides on the mRNA called the Shine-Dalgarno sequence. This is it. AGG, AGG. It's made only of purines, so structures with 2 rings, nucleotides, and it binds to a complement in the 16S region, so of part of this small subunit that is binding a match, a complementary match to this sequence. When this happens, we're going to take a tRNA, specifically FMET, and we're going to bind it to the start sequence right here on the mRNA. This stabilizes it in the P site so that it doesn't wobble out and fall. We kind of plug it in, bind it in, and then click in the tRNA. We hydrolyze some GTP, and that causes these initiation factors to fall off. We can no longer initiate. We already started, so we don't want to start again, and this prevents this falling off of these, prevents the process from restarting while it's already going. Apparently made it all the way through. The initiation complex is now ready to start, and what this means, this initiation complex is where it's all put together. So when the small subunit and the large subunit close around the tRNA that is starting the process with the mRNA already inserted, we have an initiation complex. That's a functioning, working ribosome. It's doing its thing. Elongation factors, EF2 here, TU, bring in a corresponding tRNA to bind to the codon that is now trapped in the A site. We bound our first part in the P site. We are holding on. This is a met, an F met. We then have the next codon in frame is UUC. The elongation factor brings in a tRNA that matches UUC. That means it has the anticodon AAG, maybe with a little wobble. It has its own cognate amino acid here. We have, some, goodness gracious, some GTP that hydrolyzes this, and we bind this new tRNA to the A site. This new tRNA in the A site then forms a peptide bond. I'm going to go back here. It forms a peptide bond here. The amino acid between the one in the A site and the one in the P site are joined together. Everybody slides over, and I've never been able to do this not in animation, so I've included in my set of links in Canvas an animated version of this. This is really hard to explain with words, but really easy to show. This is the second tRNA that came in. It is still holding on to its amino acid, but it is also now bound to the amino acid that was on the initiation tRNA. We are starting to create a peptide bond between them. This will slowly grow out this part of the ribosome. This P site is always going to be holding on to the polypeptide. This is facilitated by the 23S region of the ribosome. It's a ribozyme. This part of the ribosome has catalytic function on its own. If you do biochem or evolution of the biochem in living organisms, one of the things you'll talk about is the fact that we think RNA is one of the first things to show up on Earth, well before DNA. That part of the reason we think that is because RNA itself can act as an enzyme, whereas and an information molecule, whereas protein or DNA doesn't have that versatility. This is a good example of that ancient mechanism at work. Here we have an information molecule that is made of RNA and also a catalytic molecule made of RNA working together. This is different. That is different than what you learned in Bio 1, I am sure. You may have learned the enzyme that we thought did that. We do not think that's true anymore. As we shift over one more time, we translocate through the ribosome. The uncharged tRNA enters into the E site and then falls out. This is the exit site. The polypeptide remains in the P site, and the new codon is revealed. This is now, the reading frame is here. And so the elongation factor brings in a new tRNA that has the appropriate anticodon to the revealed codon. And we just keep doing that during elongation until we reach the end. Do you remember what the end is? No. She's eating my wires. We gotta stop. How do we stop? Oh, we have, all the way back from before spring break, we have this idea of stop codons or termination codons. There are 3 different versions of the end. These do not code for a amino acid. There is no ending amino acid. There is a beginning amino acid, but not an end. They instead bind to a releasing factor. The releasing factor triggers a series of events in the ribosome where it literally just falls apart. The ribosome itself disassembles. The small subunit and the large subunit fall apart. The mRNA leaves. The polypeptide goes off to do its next couple steps. The tRNAs all go get charged again, and we can start the process again. All right, a little bit of practice with me. I want to give you some bacterial practice with me and some concept practice, and then in the second video, we'll go ahead and look at how eukaryotes actually differ a little bit in their translation mechanisms, particularly with what happens afterwards. I like this question because it asks you to think about what if something goes really wrong. So we have an amino acetyl tRNA synthase. It has this amino acid on it, valine. It's supposed to load this onto the tRNA that it binds with and create this charged tRNA. But instead it's loading this glycine. So it's loading the wrong amino acid. What's going to happen now as we translate in the ribosome the strand of mRNA into a polypeptide? So I'm going to pause for a second and give you a chance to pause me. I'm not going to record 5 minutes of silence. Okay, going on. So everywhere there was supposed to be a valine, we're going to put a glycine. And we didn't mess up the other tRNA synthases. So we're still putting the correct one when it's called for. We're just also putting the wrong one every time we're supposed to be putting a valine. I think when you think about this, it's very logical. I'm substituting the wrong amino acid. This would be a pretty catastrophic mistake, I feel like. Like, this is a problem inside your cell if your amino acetyl tRNA synthases mutate. Practice with me too. Bacterial translation initiation, the mRNA, what does it do? Going on. If you have, pause me if you still need time. So the Shine-Delgarno sequence, which is unique to bacteria, is going to go through base pairing with the rRNA, and it orients the mRNA in the right location. That allows the start sequence to pull into the P site, but we bind to this sequence first, made only of purines. With that, I will move on into the next part of the lecture. I'm going to break the video there and give you a chance to get a sandwich or, I don't know, translate some on your own, take a nap. And then I'll come back and we'll do the second part of this. Right? I'll see you in the next video. Bye-bye.